Okay, let's start. All right. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Niao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you for joining us, the 48th seminar, and I call it a DOE JGI Day. Today's seminar is very special to me and to us. Tomorrow, August 26th, is our 1-year anniversary. We had only 4 times a break during the last year due to Christmas break, I believe, Thanksgiving and AICHE meeting week, having 1 seminar per week. And it has been my tremendous honor and privilege to see your excitement and support for 1 year. And I am scheduling for the 2023 season. And I hope to continue to chair this seminar in the following years. And then if you are interested in speaking, please let me know by sending email to me. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Nigel Mouncey. He is probably he doesn't need any introduction because we all know him. He is the director of DOE Joint Genome Institute called JGI. As you may know, DOE JGI is hosting a JGI meeting next week with many amazing speakers, including Nobel Prize winner, I believe. Importantly, he was a SynBYSS, the NSF Center, Synthetic Biology Center. He was a SynBYSS member and an industry mentor of one of my best students, Tatenda Chopra, guiding my student during his PhD training. Tatenda later joined Pfizer and developed the COVID vaccine production process. So I thank Nigel for his insightful, dedicated mentorship, teaching my own student about the industry and beyond. So Nigel, the virtual podium is all yours, and thank you for your time and continued support for young researcher and synthetic biology community during this also busiest time of the year due to the meeting you currently chairing and hosting. Also your continued support to the community by synthesis, service, and also sequencing service, etc. So thank you so much.
Great. Well, thank you, Taesok, for the very kind introduction. And hello, everybody. Taesok, firstly, congratulations on a 1 year of this amazing seminar series. It has really been terrific. I've watched some of the videos. Just wonderful to hear from so many talented people, both early stage in their career. We've got a bright future ahead of us, I think, in synthetic biology. And, obviously, the more established folks like myself, it's always good to hear from them what they've been up to in the last, recent times. So, I know you're all here to hear Kaushik today, so I'm going to take a few minutes just to talk a little bit about my synthetic biology journey. So I started out in the UK as a graduate student, and, it was my first foray into microbial genetics, trying to dissect a molybdenum uptake system for Azotobacter vinlandii. And that, sort of gave me a nice grounding in, the tools of the time, which, is best part of 30 years ago now. The toolbox looked very different to what it is today. I then had the, really wonderful opportunity to come to the United States to do a couple of postdocs in Boston and in Texas. And, that sort of got me on a track where I thought I would go into academia and pursue an independent, sort of more independent research career, but my career took a different turn. I ended up going into industry, firstly to work at Roche Vitamins to engineer Bacillus subtilis to improve riboflavin production. And those of you that work in the Bacillus community, know that this is a pretty friendly organism in terms of being able to genetically manipulate. When you go to work with real production strains that have been through many rounds of mutagenesis and other, genetic engineering tricks and things, these organisms don't behave like the wild type. Okay, and it took us a while to, really build up some tools and some methodologies in order to be able to create strains. And and of course, we didn't have a CRISPR toolbox at our hands then, but we used a technology called long flanking homology PCR that we built into sort of more of a modular system. I actually like to think of this a little bit like synthetic biology. We were able to do designs and build parts and then go through this building modules and do this all by PCR. And then, would then use phage transduction to get this into our production strain. And we made many thousands of mutants this way. And it was really fantastic to really be able to survey metabolism and really try to find, new ways that we could improve riboflavin production. Unfortunately, we were not successful with all of this work, but it gave me such a great knowledge about biology and metabolism in particular. That, has carried through all of my career. And I still get people today saying, why do you know about this? And I go, well, we worked on it, 20 years ago, 25 years ago. Now, I then moved, we changed company got acquired. And also that came with a move to Switzerland to work for DSM, who, at that stage were life sciences and materials, and now moving to much more completely exclusively into life sciences now. But again, sort of, using similar technologies, techniques in order to improve another route for vitamin production. And then I moved back to the United States to Dow AgroSciences to lead our efforts to improve spinosan's production by an organism called Sacropolyspora spinosa. And really there, we did a lot of work with traditional mutagenesis, lots of fermentation development. And we started our journey really to say, let's build a dedicated synthetic biology platform for this organism, for the work we were doing. And I've got to say, it was very hard. It was very hard to build this up. Again, an organism that's not particularly genetically tractable. So you're going to go back and look for promoters and reporters and all the things that, if you work with an E. coli or a yeast, you have you can pull those off the shelf, right? Those don't exist for those organisms. A lot of them that you can pull off the shelf don't work in that organism. So, there's a lot of development around that. But I think, for us, again, it was when you work with filamentous organisms, that presents another challenge in being able to get DNA in and being able to get to kind of more of a single cell level, lots of protoplast work you have to do. And that, influences how you build out your automation. And so we spent a lot of time working with automation engineers to try to really get these protocols scalable. And we were, I would say, relatively successful. By the time I left Dow, we had a pretty good platform. We were able to start to do the work we wanted to do, but it did take us a long time to do it. But there was another part of my, sort of career journey working very closely with our production facility, very large scale fermenters, 200,000 liter, multiple of these, working with the business teams in order to see how the R&D work we were doing would really translate into scale and into product, and getting that out into the hands of farmers, in particular for the insecticides we were working on. And then tying that back the other way and saying, well, what are the needs from the customers? Well, that is performance or cost or whatever. What are the business needs? How does that influence R&D? So there's sort of, two-way movement going from, the commercial side down to, getting in the lab and actually figuring out some strategies and things. Working with a big company like Dow that has multiple business, or at that time had multiple businesses before the merge and split with DuPont, was a great opportunity to work with other business units as well. And so we had a fantastic opportunity to work with LanzaTech on a really exciting project with some of our platform chemical folks. We worked on project on cosmetics with some people as well. And so it was really nice to sort of go across sectors to understand a little more about what the challenges and opportunities are in those sectors, and then how that comes, gets down into the lab, and how you can say, well, okay, what organism do I use? What pathways, should I choose? And then how to sort of engineer that, and how to boost that up. And again, not quite as easy in some of these non-traditional organisms. But again, as you go through your career, you do assemble a lot of knowledge, and the problems become a little more tractable as you can start to apply things that you've done in the past. And there's no guarantee they'll work, but at least you've got some experiences to draw from. And then over the last 5 years, I've had the really great honor and privilege of leading the Joint Genome Institute, which is a national user facility. It's open to everybody. We offer up sequencing capabilities and DNA synthesis, synthetic biology capabilities, metabolomics, and some really advanced technologies that allow people to look at transcriptional factors, to build out gene regulatory networks, to obviously play around with biosynthetic gene clusters, which is a program that I'm leading as part of my work. And a lot of data, and those of you that have had I think the opportunity to go through our data systems, hopefully have found some good use and value in the very large repositories that we have of genomics data for microbes and plants and fungi, algae, along with a nice suite of tools to analyze that data as well. It's our 25th anniversary this year. Behind me is a picture of a brand new mural that we got painted in honor of our anniversary, that really, reflects the 25 years of JGI's work. And as Taysok mentioned, next week, the week of August 29th, we are hosting a hybrid annual meeting. We've got just over 500 registrants for that meeting, and depending on when this goes out, you may still be able to join us virtually for that meeting. But we will be making, videos of some, if not all, of the talks available through our YouTube channel. So I will just leave a little perspective on synthetic biology. I mean, this is a very exciting time for biotechnology. The pandemic has brought into focus genomic science, and I think science in general into, I would say a new light. And I think people have been, see the value that science and genomics and synthetic biology can bring to the world, and in order to solve problems. And we're seeing now, I think, this really great kind of I would say renewed movement now of biotechnology and what biotechnology can do. Obviously the vaccines, especially those mRNA vaccines, it's an incredible success story. It really is, and remarkable. And I think now with, the tools that we have available, the knowledge we have available, that we're going to see many more really exciting products coming out of biotechnology in the next few years. And so, this is a great time to be thinking about career in biotechnology, whether, continuing in academic space. There's a lot of biology we don't know, we don't understand. There's a, tremendous amount of, fundamental science that has to be done that then can fuel the applied side. So, as you're thinking about careers, please, I think this is a great path. It's a great time. It's an exciting time. So I will leave it there. If you have questions for me afterwards, I am pretty easy to find on Google and things, but my email is nmouncey, M-O-U-N-C-E-Y, @lbl.gov. So, and feel free to please send me an email. Great. Thank you.
Wonderful, wonderful. So, you know what? When I heard, your entire story, I feel like you are living history of synthetic biology or whatever engineering biology. Because I also remember when I start my research late 1990, I think 7. At that time we don't have any mini prep kit, so we need to do phenol extraction, everything for 3 hours to just get E. coli DNA plasmid. And then we're talking about homologous recombination. Oh my God, that's nightmare. And then, but we are now have lots of tools, CRISPR, DNA synthesis. So we need to do better, of course, to move the technology forward. And also, I love what you said at the beginning. You learn a lot from failure, your first company, I believe. That's my advice to every single young person, because I always tell people saying, fail miserably when you are young. In that way, you could recover from that failure and then you will learn a lot. That will be probably the best lesson to anybody, because you learn a lot from the failure. And then fail when you are old, recovering is not that easy, but when you are young, you could recover and then do it better. So you gave really nice real time or real history of your kind of experience, and that is brilliant. So I love it. So thank you so much.
Thank you.
Yeah. All right. So okay, so now the main speaker of today with the longer introduction, and Dr. Kaushik Venkatesan is a recent chemical engineering PhD graduate, now joined Genomatica, but his interest in using a data-driven approach to advance systems biology research. Specifically, he is interested in using cutting-edge techniques in machine learning to reinforce mechanistic models of metabolism, gene regulation, to better predict strategies to engineer organism for the production of therapeutic and industrially relevant compounds. So I love, what he is doing, because the current keyword, one of the keywords is of course machine learning, and then combining that with mechanistic model, because with that we could learn some probably governing rule in biology, hopefully later on. During his PhD training, he worked on developing an array of technologies to develop microbial chemical production platforms, including automation-assisted high-throughput experimentation platforms, metabolic network-based computational algorithms, and gene regulatory device. He has also taught several courses that discuss the fundamentals of machine learning, dynamical models, and their application in biological context. And he's interested in academic position, but currently as an engineer, he wanted to sort of have experience in industry, and then wanted to go back to academia. In that way, as an engineer, he probably have the better broad perspective in this kind of field. So with that, Kaushik, thank you so much for your time today, and please take it away, and it's all yours now. Thank you.
Thanks, Professor Moon, for that introduction, and thanks, Dr. Mouncey, and everyone for joining us, joining me today on the seminar. Let me share screen. I hope my screen is visible.
Right now, the slide is visible, but slide mode. Okay, now, yeah, you need to do that. Okay, now better. Wonderful.
Great. All right, so thanks again, everybody, for joining me today seminar. Today, so before I go, I'll introduce myself. I'm Kaushik, recent graduate from Professor Mahadevan's lab at the University of Toronto. And today I thought I'll discuss two of the projects that I worked on during my PhD. And the title of my PhD thesis was Design Principles for Engineering Dynamic Control of Metabolism. So this talk will involve a lot of dynamic control of metabolism principles. So just some quick context on what I'll present today, quick background on engineering microbes for chemical production and dynamic control. Then I'll talk about this project where we demonstrated the advantages of dynamically controlling metabolism in bioprocesses. Move on to an actual implementation of dynamic control, figuring out how to improve genetic control devices in order to implement dynamic control. And then I'll conclude with some thoughts on what future work can be done in this space. Quick background on why we wanted to study dynamic control of metabolism in Krishna's lab. So fossil fuels are still an important source of many of the important chemicals that we use as commodities or industrial raw materials. While a lot of the fossil fuels or crude oil that we produce goes into transportation fuel, nearly 15% of every barrel of crude oil is used to make these other chemicals, industrially important chemicals that we use every day. The problem with conventional modes of chemical production, we all know, is that typically high emissions are involved and they use a non-renewable substrate. And there is a need to develop more renewable alternatives to conventional modes of chemical production. And microbial chemical production is a relatively benign alternative since it uses glucose or other renewable substrates as feed. And when these are fed to a microbe of interest, using their own metabolism or enzymes introduced into them, they can manufacture the product of interest. And some organisms can even use CO2 or CO2 derivatives directly. That's a big focus area right now. And like I said, this is less process intensive than conventional modes of chemical production since microbes grow at relatively benign process conditions. And also another advantage of this whole process is that because metabolism is so diverse, we have a huge array of products that we have access to. But the problem in these processes is that it's not always economically feasible right out of the bat. And there are three metrics that are typically considered industrially relevant when we're determining the feasibility of these processes at an industrial scale. So these are what I like to call the TRY metrics, the titer, rate, and yield. Titer is the concentration of the product at the end of fermentation. Rate is the speed at which the microbe is able to produce the chemical. And yield is the amount of chemical product that the microbe can produce per gram of substrate fed. Now, base strain undergoes several rounds of the design, build, test, learn cycle before we can come up with a scalable production platform. And this process basically accounts for a lot of the cost involved in developing strains. And my PhD, I wanted to work on a fundamental project that would help enhance these TRY metrics. And one of the avenues that interested me a lot was this concept of dynamic control of metabolism, which has been touted as a way to enhance these production metrics. So I'll go into the first project that I'll talk about today with some background on what dynamic control is. But before that, again, background on engineering microbes for chemical production. So wild-type organisms take some substrate, extract energy, and release some byproducts. They use it to grow and replicate. Now, the metabolic capabilities of a microbe can be mapped using its production envelope, which we can get from its stoichiometric, its metabolic model. And given a metabolic model of all the reactions that occur inside an organism and a rate of feedstock uptake, we can basically get the maximal production rates of our products of interest using these production envelopes. So what this production envelope describes is at each growth rate possible for the microorganism, how much of product or what is the maximum amount of product we can extract from it. And typically a production envelope looks like this, where when the microbe is growing at its maximum capability, it's not producing any product. For most products, for a lot of products, this is the case. And when we're producing the product at the maximal possible levels, there is no growth. Now, microbes, wild-type microbes operate at this point because they've evolved for faster growth and better substrate uptake. Now, traditional strategies for chemical production involve deleting a reaction or entire pathways to stop carbon flux towards undesired pathways and redirect it towards our pathways of interest so that we can produce the chemical of interest. And these processes are implemented as one-stage production processes where the microbe both grows and produces product at the same time. Now, these one-stage processes are thought to offer higher yield. Now, an alternative to one-stage production process is a two-stage process where we decouple growth and production into distinct stages. Now, what happens is we start with a stage of with the growth stage where the microorganism grows at maximal levels, and then after some time we switch its metabolic state and activate production pathways and deactivate growth. Now, all of the cells that we accumulated in the first stage are now able to produce chemicals at higher quantities in the second stage. And this is thought to offer higher productivity because we have accumulated a lot of biocatalysts in the process. So this is good, but one problem is that substrate uptake rates during the stationary phase of metabolism, or when we stop growth, basically drops. And low substrate uptake implies we can't produce as much product as we intended to in the production stage. And this called into question this concept of high productivity from two-stage processes. And one of our collaborators from Germany, Professor Klampt, did this study where they examined whether two-stage processes can outperform one-stage processes. And they concluded that if substrate uptake rates are impacted, the productivity in two-stage processes is actually lower. So we wanted to look at in what cases two-stage processes can outperform one-stage processes, and are there newer kinds of two-stage processes where we don't need to cut off growth completely, but throttle growth at intermediate levels so that the microbe can still produce chemicals and grow at the same time in its second stage. So in order to access all of these intermediate operating points, we came up with a computational algorithm to explore all of the operating points and compare two-stage processes and one-stage processes. So in this computational model, computational algorithm, we take as inputs the approximate substrate uptake characteristics of the microbe, get a metabolic model for the organism of choice, and generate a more realistic production envelope. The previous production envelope, we saw that maximum product flux was at zero growth, but if we account for substrate uptake limitation, it comes down a lot. So once we have this more realistic production envelope, and given some fermentation parameters like initial substrate concentrations and initial biomass concentrations, we simulate the fermentation process using a dynamic flux balance analysis, and using an optimization formulation where we give as the objective function some combination of the titer rate and yield. We can obtain which what the maximum value of this objective function will be given a set of 2 operating points on this production envelope. So doing this for different operating point combinations on the production envelope, we're able to generate a landscape of this objective function, which tells us for every combination of the operating points, what the maximum value of the objective function can be. And again, this objective function will be some combination of the industrially relevant parameters, titer, rate, and yield. So as a test case, we wanted to use this combinational algorithm to first maximize the productivity of lactic acid in E. coli. So again, the objective function for this analysis is productivity, and all the operating points are represented by their growth rate in each stage. So all one-stage processes in this plot are represented by the diagonal, since we have the same growth rate or the same operating point in both stage 1 and stage 2. And a one-stage process with the maximum productivity occurs somewhere around here, where the organism is both growing and producing product at the same time. A conventional 2-stage process comes somewhere around here, where we're growing at the maximum rate in the first stage, but completely cutting off growth in the second stage. And this is what our collaborators observed. But we have this whole area of high productivity, and the best 2-stage process occurs somewhere around here. And these have higher productivity than the best 1-stage process. And these have maximum growth in the first stage, and intermediate growth and production in the second stage. Now, we then expanded this for all 70 exportable products in E. coli's latest metabolic model, provided a snapshot here, but the trends remain the same. So we see the highest productivity for the green dots here, which are basically the best 2-stage processes. The best 1-stage processes have lower productivity than the best 2-stage processes. And below both of them come the conventional 2-stage processes, which suffer because of lower substrate uptake rates. Now, in an industrial setting, productivity is not always the objective. Yield is also important. So we perform the same analysis for a second objective, where we try to maximize productivity given that the yield has to be greater than 70%. We observe the same trends. And as the final objective, which we thought would be the most industrially relevant objective, we tried to maximize yield given a constraint on productivity. We found that surprisingly, 2-stage processes do offer maximum yield if we set that constraint of productivity there. So this was really nice, but we wanted to go further and figure out how we actually can go from these growth stages to production stage. So we looked at the flux changes because we're using a metabolic model, we're able to look at what fluxes need to change between the growth and the production stage. And we grouped them by the reaction subsystem that every reaction is involved in. And we did this for all the 70 exportable products in E. coli's metabolic model. And we saw that most of the reactions fluxes are reduced by the same amounts as growth rate, which is a good thing. As growth drops, the fluxes through these reactions also drops, about 60% in the second stage. But there are some pathways which where the fluxes are increased and decreased as a whole, and specifically the TCA cycle, where the reactions are turned off or reduced by 100% for a lot of the reactions. Glycolysis, where the fluxes are maintained, even though other reaction fluxes go down by 60%, which is the reduction in growth, glycolysis still maintains its flux, which makes sense. We need to uptake substrate and process it in order to provide the precursors for the product of choice. And most interestingly, it was the pentose phosphate pathway, which was divided between upregulations and downregulations, the reds and blues here. So the pentose phosphate pathway acts as a key node, which basically determines what precursors will be produced for each product. So that's a really nice thing that came out of this analysis. And then we looked at the reactions whose fluxes are modified for a majority of the products that we analyzed, because we thought that these would be key reactions that we need to control for several products. And we grouped them based on whether they needed to be turned on, off, upregulated, or downregulated in the second stage. So going from the growth stage to the production stage, which reactions need to be turned on the most, which reactions need to be modified for the most number of products. And some clear important reactions emerge here. Specifically, these ones, we analyzed for 70 products, and for nearly 60 of them, these reactions show up. And if we look at what those reactions exactly are, the ATP-dependent glucose uptake system is activated for a large number of products. Well, conventional modes of glucose uptake involves the use of PEP. In our second stage or in our production stage, the ATP-dependent system becomes more active. And similarly, these two reactions, S6PA and DHAP-T, which are involved in central carbon metabolism, are turned off for most of the products. And these two reactions together use up PEP. And so consistently, we saw that reactions that or strategies that conserve phosphoenol pyruvate are enriched in this analysis. Other important trends that emerged were that, again, the TCA cycle reactions are downregulated or turned off, and the pentose phosphate pathway reactions are divided between upregulations and downregulations, just like we saw in the plot above. Now, having done this, when we submitted this paper, we got very interesting reviews. So we were basically asked the question, does our substrate uptake rate characteristic make sense? So we had assumed a logistic curve to model the variation of substrate uptake rates versus growth rate. And when we mined literature for the variation in substrate uptake rates versus growth rate, we saw that the logistic model does not fit the substrate uptake rate characteristics very well. And so we wanted to perform the same analysis for any substrate uptake model. So what we did was we allowed the substrate uptake rates to change from the minimum feasible uptake rate determined by the metabolic model to the maximum observed in industrially relevant, the maximal observed substrate uptake rates in the metabolic model. And we performed this analysis for all exportable products and figured out what is the fold improvement in productivity in a two-stage process when compared to a one-stage process. And we found that regardless of what the substrate uptake rate is in the second stage, there are two-stage processes with intermediate growth that offer higher productivity than the one-stage process. And we performed this for the other objective, where we constrained the yield to be at least 70% of the maximum theoretical yield. And we observed the same trend. Wherever a two-stage process was feasible, it outperformed the one-stage process. And this red region here is where no bioprocess is feasible. So if the substrate uptake rates fall into this region, we cannot guarantee a yield of 70%. So that's what the red region accounts for. And similarly, for the other case where we try to maximize yield given productivity greater than 2 grams per liter per hour, we saw that the maximum yield always occurred in the 2-stage process optimal region. So conclude this study quickly by saying what we did. We developed a computational tool to predict optimal bioprocesses and metabolic phenotypes, and showed that 2-stage processes do indeed show optimal titer rate and also yield for all the products that we analyze. And we produced, we formulated new kinds of 2-stage processes which have partial growth during the 2nd stage. And we thought that the production stage fluxes revealed some common patterns, like reactions that occur consistently across all products, which give rise to this possibility of a modular production platform. Now, having done this project, we wanted to look into how we actually implement such dynamic control of metabolism. And one tool that we were interested in is this concept of a genetic toggle switch, which came out of Professor Jim Collins' lab at MIT. So this is widely considered to be one of the pioneering inventions in synthetic biology. And I'll quickly go over how it works. So it's a bistable motif, meaning that it takes one of 2 stable steady states. So its architecture, it has 2 repressors which repress each other's production. So when the switch is in its θr state, that is when we add IPTG and remove LACI's ability to repress its target promoter, θr is at its high state, and θr goes and binds to its target promoter, which controls LACI production. And even after we remove IPTG, the system remains in the same state. It doesn't start producing LACI because of this repression. And now, if we add ATC, which sequesters θr, the switch can now produce LACI, and this LACI will go and bind to its target promoter, which prevents θr from being produced. And after that, we can remove ATC again, and the system remains in its same state. So this sort of functions as a primitive mode of cellular memory, because the cell remembers the last inducer it was exposed to. However, in using this system in some of our studies, we found that the system lost bistability, that its ability to retain its steady state even after the inducer is removed. It lost that ability. And we also found that the switching speeds between the 2 states are kind of slow, which made scale up of these scale up of systems that use this tool difficult. So we wanted to look at the system in much more detail. So and specifically analyze which parameters influence the bistability and switching speeds. So we took a dynamic model of how the switch functions based on mass action kinetics. And in this model, we track the repressor concentrations, LACI and θr, and see how, formulate how they vary with respect to time as in ODE. And we parameterize the model with the protein production rates, basically transcription translation rates, the repressor efficiency, which is how well the repressor is able to bind and prevent transcription from its target, and also the degradation rates of the proteins involved. Finally, the cooperativity, or the number of LACI monomers or θr monomers that have to come together in order to repress their target promoter. And specifically, we were interested in these 2 parameters, protein production and degradation rates, because they're easily engineerable for E. coli. E. coli-based systems, and we wanted to look at two kinds of simulations, basically bifurcation analysis and dynamical simulations, and in order to study how these two parameters affect the switching function. And when it came to switching function, we were interested in two functional metrics. One was the robustness of the system to different protein production rates. So this we obtained by performing what is called continuity analysis. Basically, we figured out through bifurcation where the system goes from bistability to monostability. So a bifurcation analysis gives us the list of parameter values where the system remains bistable. So this is a simple continuity plot where we show the range of parameter values where the system remains bistable. And all parameter values within this plot are where the system remains bistable. So we formulated this robustness metric as a fraction of the area bound by this curve, which basically tells us how much we can perturb the protein production rates in the system before it becomes monostable. And we did this for different parameter combinations spanning the entire range of feasible parameter values for the system. And we came up with a robustness landscape where we analyzed how this robustness metric changes given changes in the degradation rates of the two proteins. So one thing that came out immediately was that as the degradation rates of the protein of the repressors involved in the system increases, we find that the robustness metric keeps dropping. And another thing that comes out is that balanced toggle switches that have the same degradation rate on both the repressors are more robust than the ones that have largely different degradation rates. Now, having studied the robustness, we also wanted to look at the speed. So using dynamical simulations, we were able to look at how much time the system would take to go from one state to another, and formulated a speed metric. And here we observed something that was expected. As we increase the degradation rates, the speed of switching keeps dropping, becomes faster and faster. And to compare the speed and robustness metric more directly, we plotted the speed versus robustness, separating each analysis by separating each simulation based on the ratio of the degradation rates that we use. And this way, the trade-off between speed and robustness becomes more apparent. As we increase the degradation rates, the robustness metric drops while the speed increases. And we also find that there's an optimal ratio of degradation rates where we get the maximal robustness for each switch speed. In this particular case, it's 1, but we did the same analysis for all feasible combinations of parameter values, and we found that the degradation rate ratios that balance out imbalances in other parameters in the system basically lead to optimal robustness for a given switching speed. Now, all of this is computational dynamical modeling work. We wanted to verify the system experimentally and see if the speed robustness trade-off actually comes out in experimental systems. But when we tried implementing it, when we tried analyzing it on the original toggle switch, found that the repressor and the reporter are decoupled. So it's not possible to accurately track how much of the repressor is in the system. So that made an evaluation of the speed metric impossible. So we came up with the strategy to couple the repressor and the reporter, fuse them with linkers, and analyzed all possible combinations of the repressor and the reporter. That is the repressor at the N terminus, or the C terminus, and figured out which combination of the repressor and the reporter would lead to repression characteristics similar to the wild type repressor. We found that TET-R at the N terminus and LAC-I at the C terminus gives us repression characteristics that are similar to the wild type repressors. Use those to build a fusion version of the toggle switch. And then we wanted to see how the protein production rates and degradation rates will affect the switch functioning. So we varied the ribosomal binding sites, and using Professor Howard Salas's RBS calculator, designed several RBSs and used the native SSRA degradation system in E. coli, and obtained tags that offer graded degradation of the target proteins. So we targeted 5 different levels of protein production and 4 different levels of degradation, giving us 20 constructs each for the GFP LAC-I and the TET-RM cherry. And when we characterized each variant, we determined the steady state levels of GFP and M-cherry, which are proxies for the repressor concentrations. And what we saw that while degradation tags function as expected, that is, as we progressively increase the strength of the degradation tag, the steady state levels drop. The RBSs, so some of the RBSs did not show high variability, particularly for TET-R and cherry. So we decided to pick 10 constructs from each repressor reporter fusion that gave us the highest variability in order to build a combinatorial library that is of GFP LAC-I and TET-R and cherry. So since we had 10 from each, we got a 100-member representative toggle switch library. Now, next, we analyzed this toggle switch library using a high throughput platform that we developed. And first, we wanted to look at the switching speed. And to look at the switching speed, we again considered the fluorescence from the reporters. And there are 2 speeds that are important here. One is the switch from LAC-I to the TET-R state, that is when M-cherry is high and GFP is low. And the other one is the TET-R to the LAC-I state, that is when M-cherry becomes low and GFP becomes high. And the initial fusion construct based on RBSs and no degradation from Professor Collins' lab, that gives us switching speed somewhere around 4.5 hours, or 4 hours either way. And this is a plot of our entire 100-member library system, where we saw a large variation in the switching times. So adding the degradation tags did indeed change the switching time a lot. And we've colored, based on the rate of degradation that we expect from each degradation tag, we binned the different constructs into whether they offered low, mid, or high degradation rates for each of the repressors. We found that the TET-R degradation tags seem to have a higher effect in controlling the speed of switching. And we see that the constructs with the high degradation rates are typically the ones that switch the fastest. That is, they take the least time to switch between the 2 states. But there were several constructs with intermediate degradation rates, shown in gray here, that have very slow switching. And we wanted to look at why that was. And so we looked at the growth rates of the host organism for these constructs. And we found that these constructs that have intermediate degradation, but slow switching, are usually because they have very slow growth. So growth rate also, the growth rate of the host also impacts the switch functioning. And that is one parameter that we need to consider in the future. And this we observed for both the TET to LAC switching and LAC to TET switching. So having observed the effect of the degradation tag on switching speeds, we want to look at how it actually affected the robustness of our switch. So we separated both repressors based on whether they offered low, mid, or high degradation. And at the lowest levels, and we figured out what percentage of replicates actually remain bistable in the system. And at the lowest degradation bands, most of the constructs remain bistable. And as we progressively increase the degradation rates, the number of constructs or the percentage of constructs that remain bistable becomes smaller and smaller. And this we observed for both the TET and the LAC degradation rates, with the number of bistable constructs becoming smaller and smaller. And even for combinatorial variations, we observed the same trends, with the least number of bistable constructs coming from the highest degradation bands for both repressors. And to compare the robustness and speed more directly, we put them on, we put all the strains that we analyzed on one plot, colored them based on whether they're bistable or not. And if we look at what percentage of constructs are bistable for each switching speed band, find that at the fastest switching speeds, we have very few constructs that are bistable. And as we progressively look at slower switching speeds, we find that more and more constructs become bistable. And this is experimental evidence that the speed robustness trade-off that we observed from our model actually holds up in experiment as well. And in order to determine which constructs to use for future evaluation, we also needed to look at growth rates, because we don't want constructs that lead to a huge impact on the growth of the host organism, because that also impacts the industrial relevance of these constructs. So we identified one construct that had fast switching speeds, remained bistable, but still did not impact the growth rate too much. And we're currently using that to in order to try and produce, improve the productivity of lactic acid in E. coli. Concluding this study, so we developed a dynamical modeling framework to characterize the stability and the speed of genetic circuits, and looked at which parametric regimes resulted in optimal function of the switch. And we developed a modified version of the toggle switch that allows us to track the switch, the repressor concentrations of within the switch easily. And we established a trade-off between the speed and robustness of the toggle switch, and found that the growth rate of the host also impacts the switch characteristics. So in I'll just quickly conclude these two studies and talk about where the future lies. So from our first project, we showed that dynamic control can be used to enhance chemical production, and picked the toggle switch as a tool for that we wanted to implement, and we wanted to improve the characteristics of the toggle switch. And we initiated a design, build, test, learn cycle, where we first used the model to design optimal constructs of switches, built all of them using ligase cycling reaction, evaluated our 100-member library with experimental replicates on our in-house automated strain evaluation platform. And we learned that there is a functional trade-off between robustness and speed, and that growth rate also impacts the functioning of the switch. So from one cycle of the design build test learn cycle, we got an optimal switch construct, and which we plan to use for further evaluation. And as a recommendation or avenues for future work, I see that the number of, in the first project, the number of fluxes that need to be perturbed to go from growth to production are very large. So working on reducing the number of fluxes that actually need to be changed to affect the growth to production state change. And also explore the concept of universal production platforms in more detail. And finally, evaluate the effect of growth rate on genetic circuit function more thoroughly in the future. That I do want to thank a lot of collaborators over the years who helped me in completing my PhD, and also in working on different projects that I talked about today. And yeah, thank you.
Okay, so thank you so much. Wonderful talk. So Nigel, do you have any challenging question or comment?
Absolutely terrific. Both parts of your talk I really enjoyed. So I have a question for each part. So let me start with the first one, the two-stage process. One of the things that we found running industrial fermentations was that it would be really advantageous for us to maintain maximum productivity for as long as possible, so that we didn't have to go through, that biomass build and just maintain that sort of production time frame as long as possible. Is that something you looked at? I wasn't quite sure from what you described, whether you looked at kind of just strategies for maximizing productivity from what you found for as long as possible.
So one of the things that the computational framework does is figure out at what time the switch needs to happen from growth to production. And we do find that, I don't have the exact numbers from the computational framework exactly here, but like you said, the longer we have the microorganisms in their production stage, the better for productivity. But actually sustaining the production stage is indeed a challenge, like you said, and that is something that we need to look at in future, building more robust microorganisms to tolerate the production stage.
Yeah, and your sort of partial growth thing. So one of the things that we found was that you get this decrease coming down in maximum productivity. I mean, so we found ways to sustain it for let's say half a day or whatever, but then it would come down. And we looked into things like product tolerance, etc., and didn't rule that out. And something there's something in metabolism, and I would say for at least I would say most of the fermentation processes, which were quite distinct in a way in terms of what we were making, this same thing came up. So it almost suggested to me that there's something fundamental in metabolism that you need to replenish, whether it's energy or whether it is cofactors or something that if you do go back and actually say, well, let's have a bit of a growth stage again, can you then go back to a point where where you even can just sustain, let's say, half productivity even would work if you can sustain it long enough?
Yeah, that is indeed a challenge. And I feel that for different processes or different products, maybe it's different. So if we're producing something that's extremely toxic to the cells, we need to account for the fact that the cells will basically die during the production stage if the toxic product accumulates a lot. And like you said, there may be cofactor limitations. So those are things that needed to be added to this computational framework to make it more robust.
Yeah, that was really, really, really interesting work. And then my sort of question for the second part is, really elegant work of building on Jim's, original switch there. Was all the work done on with did you integrate these various switches into the genome, or were they on plasmids?
Right now, we did a plasmid-based system, so they're all in plasmids.
Okay. Do you think there's any influence on plasmid copy number stability on
Absolutely. So,
data?
yep. That definitely impacts the switch function. And I feel that plasmid copy number is a significant burden on the cells. And that is one of the aspects that led to the slowdown in growth in some of our constructs. And there is definite variability in plasmid copy number between our strains as well. So that is something that can make the model better. But the model that we used is sort of a lumped model that, so the plasmid copy number affects the transcription translation rates and so on. The one thing that we did not account for in the model is the growth rate. And that came out during our experiments. So that's where we're thinking of improving this model in the future.
Okay. Yeah, great. Yeah, no, very, very interesting work. Yeah, thank you.
Wonderful. So actually, the Nigel asked some question I wanted to ask. So I wanted to check though whether there is any question from audience. Okay, I didn't see any. Okay, I'll ask question. So first one is, this is brilliant talk. I love it. And then actually one comment on the second part is, you use the IBS calculator. I love the calculator a lot, but you need to also remember the calculator has limited accuracy. I would say probably twofold difference would be kind of error range, but you design somehow 10-fold, 24, and 34. Probably that is probably one of the mistakes, when you are kind of doing the experiment. So I guess you need to learn from your mistake and for the future, direction. My broad question is very challenging because I also challenged Jay Kiesling during his keynote talk during SynBYSS conference this month. And the question is, he demonstrated beautifully, I would say the best paper ever in this year, 30-step pathway to make anti-cancer drug. The title is tiny, but I would believe that is probably one achievement this year. I love it. But when I heard his talk, I still kind of believe many different, optimization, basically troubleshooting, and then systematically his team did. But still, I kind of sometimes feel like this is still art, even after 30 years of metabolic engineering. So you kind of show now promising future direction to me, that is computation model-based metabolic engineering. But still, you need to consider many different things because Ford engineering is not easy. So my question, the challenging question is, what should be done in terms of make your model more predictable in the future? And that means what is current remaining challenge to go there?
So yeah, like you said, I feel that in biology, there's just so much variability that coming up with a mechanistic model that determines exactly what the cell will do or exactly what the toggle switch will do, it's very difficult. And so keeping that in the back of the mind, we can still use the models to give context on results and how we can improve certain aspects of cells. So in this specific case, we found that the speed robustness trade-off does exist because of the model. And we were able to observe it in experiments, but there were some outliers. And the outliers we found were because of growth rate. So I feel like model building is always an iterative process. And the more we learn, the better we make our models. But still, the way to improve models, again, is doing more experiments. And one of the avenues that I'm excited about is building hybrid models using and leveraging machine learning techniques to use the data that we collect from experimental work in order to get better insights from these models. That's one avenue that I'm really excited about in the future.
Sure. So I agree with you. What I want to see is, so for example, so without building model, based on my intuition or my experience for the toggle switch, dilution is very important, and you demonstrate experimentally, because without thinking of, without seeing the mathematics, I know immediately, because dilution is important, because the protein, the repressor should be, concentration should go down to switch fast, whatever. But what I'm saying here is, in the future, what I want to see to make the metabolic engineering more engineering or forward engineering, I wanted to see whatever model with some minimum number of experimental data set. I want to say, okay, 30 different step pathway, step 1 enzyme in the expression level should be 100, and the second one is 200, third one is 10, and so on, depending on the characteristic of the enzyme activity, whatever. Probably that might be not 10 years from now, but based on the technology speed, development speed, I believe those kind of thing is not impossible. We could see that one. I want to see that before I die. And so that's my kind of mindset, but it might be too ambitious, but it's not impossible. So that type of thing I expecting from people, from machine learning, from a lot of data set we currently generating. And that's kind of one thing I want you to think about in the future, because that I want to see and many people wanted to see. Right?
Yep. It's very interesting that you bring that up. I think Dr. Hector Garcia Martin from
I know him very well.
Yeah, so he's I think working on that, and I've read some of his papers. I think for tryptophan production, where they try to use machine learning to improve the production of tryptophan. So there is research being done in that space as well, and it's really exciting.
Yeah, he's my collaborator. We are currently writing papers together, so
nice.
paper together. So I hope to see something from him, but I also wanted to see something from you and young people. He's getting old, so but I need to expect more from young people like you, because your imagination, no limit, right? So that's what I want to see. All right. Any other question, Nigel?
No, this was wonderful.
Okay, so let me check one more time. So no question from chat. Okay, Q&A. Okay, so I believe we already passed 11, now almost 20. So I will close. Okay, so thank you all for joining and staying today. We'll meet again next week on September 1st, Thursday, the same time, the same Zoom link. We are starting a new semester with great speakers again. We'll have Professor Greg Stepanopoulos from MIT, of course, the metabolic engineering father, and Professor Ben Woolston from Northeastern University. And as usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with us. Thank you, and I'll stop recording now.